So just a little bit about Dr. Rozaini before we start. Dr. Rozaini is actually our Deputy Director at Al-Rahman Institute for Natural Product Discovery. So Dr. Rozaini as well is a senior lecturer in Faculty Pharmacy from Life Sciences Department. So her research interest is mainly on drug discovery from natural product for wound healing, malaria, and diabetes. And for today, she will share with us the nature's armamentarium against malaria. So, Dr. Rozaini, the floor is yours.
Right, thank you, Dr. Nurmala, for the lovely introduction. Assalamualaikum and good morning, everyone. The reason why I choose this topic is because my research team has quite recently involved in the anti-malaria research work. My previous works mainly focus on the wound healing efficacy of natural resources such as honey, gamet, and Gametocytes, which perhaps I can share with you in some other time. All right, so without wasting more time, let's have a look at the first slide. Today's topic is on nature's armamentarium against malaria, which is quite broad actually. And in fact, there is a lot of naturally occurring compound which has been proven to be effective as anti-malarial agent, and some has even entered clinical phase. But for today, I will only focus on the two most important anti-malaria drugs, which are derived from plants and are a vital part of our chemotherapeutic armory against malaria. So the first one would be quinine from the cinchona tree. From South America during the 19th century, and the second one would be Archemazine from Artemisia annua from China. So before we proceed to discuss the natural products, I think it's better to have an overview: what is malaria, what causes the disease, its prevalence in Malaysia, and its global burden. So what is malaria? Malaria is an infectious disease that is spread transmitted by mosquitoes, in particular female mosquitoes of the genus Anopheles. In Malay, we call it as nyamuk tiruk. These mosquitoes are known as night-biting mosquitoes because they most commonly bite between dusk and dawn. One of the distinguished features of Anopheles mosquitoes is the roughly 45-degree angle of their abdomen during a blood meal. This is actually unique to Anopheles and can be used to identify the mosquitoes. The Anopheles mosquitoes require a temperature climate to thrive, a high temperature climate to thrive. Thus, malaria is commonly found in the warmer regions of the world that are closer to the equator, including tropical and subtropical countries. The Malaria parasite, which develops in the mosquitoes, also require a warm environment to complete their growth cycle before reaching the stage at which they are ready to be transmitted to humans. I will briefly discuss the symptoms so that we have the same starting point. Classic malaria is a cyclic fever that comes every 3 to 4 days. It began with cold stage where patient feel chilly with a sensation of headache, nausea and vomiting as the temperature rises. As patient hit the fever stage, the temperature goes to 39 to 40.5 degrees Celsius. Patient become very lethargic at this point. And as the fever breaks, patient will have a cold sweat and they are profoundly fatigued. In most of P. falciparum infection, these can go to a much severe form, and the worst case being cerebral malaria, where a patient can go into coma and die. Patient can also experience severe anemia and become hypoglycemic with electrolyte disturbances, jaundice, lactic acidosis, and eventually death. The word malaria actually derived from medieval Italian for bad air, since it was assumed to be caused by intake of polluted or foul-smelling air given off by swarms. In fact, it was also called swamp fever and is one of the most ancient infections known to humans. There has always been some recognition of the association between swampy, marshy areas and the disease, although the exact roles of the mosquitoes and the parasite were not known until the beginning of the 20th century. In fact, malaria is caused by a single-cell protozoan parasites belongs to a genus known as Plasmodium. There are many different types of Plasmodium parasite, but only 5 types can infect humans. These are Plasmodium Falciparum, which is responsible for the majority of malarial death globally and is the most prevalent species in sub-Saharan Africa. Next is Plasmodium vivax, the second most significant species, and is prevalent in Southeast Asia and Latin America. It can stay in the liver for up to 3 years, which can result in relapses. Plasmodium malariae, which represent only a small percentage of infections. Plasmodium ovale, which is fairly uncommon and usually found in West Africa and can also remain in the liver for several years without producing symptoms. The last one would be Plasmodium knowlesi, which is a zoonotic malaria parasite which originally infect primates and is the most common causes of human malaria in Malaysia, especially in Sabah and Sarawak. Okay, the slides is an example of a blood smear stain with GIMSA or GIMSA stain, showing the red blood cell infected with the Plasmodium. Basically the red blood cells, this is the red blood cell, and the purplish dots inside the red blood cells are the parasites, the red blood cell which is infected with parasites. The Plasmodium has a complex life cycle. Part of it takes place inside a human host and part of it takes place inside a mosquito vector. There are essentially 5 key stages to the Plasmodium life cycle. So the first one would be when the Anopheles mosquito bites a human, injecting the Plasmodium parasites which enters the human blood. At this stage, the parasite is in a form known as sporozoid, which is long and thin and is capable of moving in between and within cells. The parasite travels in the blood until it reaches the liver. At this point, the parasite recognizes and invades liver cell where it remains for around 10 days. In the liver, it undergoes a transformation into thousands and thousands of new parasites known as merozoites. These newly formed merozoites are released into the bloodstream. The merozoites invade red blood cells and then reproduce. Each merozoite enters a red blood cell, and once inside, it grows and divides asexually to form up to 20 new merozoites. These then burst out of the cell and invade neighboring red blood cell. This whole process takes approximately 48 hours. Some parasites do not form merozoites, but develop into sexual stage of the life cycle called gametocytes. They are taken up by mosquitoes when they feed on an infected human. Once inside the mosquito's stomach or midgut, the gametocytes change into a mature gametes, which fuse and develop into an ookinete. The ookinete then burrows through the lining of the mosquito's midgut wall, where it forms an oocyte in which tens of thousands of sporozoids are formed. They burst out of the oocyte and travel to the salivary gland of the mosquito, where the cycles begins again. So most of the anti-malarial drugs targets the blood stage of the parasite's life cycle, the stage that causes illness and symptoms. However, very few anti-malarial drugs targets the liver stage. Due to the growing resistance to blood stage malarial drug, the discovery of new liver stage anti-malarial drugs is therefore both timely and important. This is the developmental stages of malaria parasite, which in this case is Plasmodium falciparum, seen in blood films of infected patients. Okay. During the earlier stage, which is the ring stage, multiple parasites are more likely to be seen within the red blood cell. The trophozoite stage is the growing stage, where the parasite within the red blood cell may vary in size from small to quite large in size. Malaria pigment is a byproduct of the growth or metabolism of the parasite, and it appears as the parasite grows. Okay, at the schizon stage, the malaria parasite starts to reproduce. This reproduction is referred to as asexual because the parasite is neither male nor female, but reproduces itself by simple division. The gametocyte stage is sexual in that the parasite becomes either male or female in preparation for the next stage, which takes place in the stomach of the female Anopheline mosquitoes. All right, in Malaysia, the history of malaria started in the 1960s when about 240,000 cases of malaria were documented across the country. However, since the establishment of the Malaria Eradication Program in 1967, numbers of malaria cases have decreased significantly. From 2013 to 2017, a total of 16,500 malaria cases were notified in Malaysia. In general, Sabah and Sarawak contributed more than 75% of the notified malaria cases in Malaysia, while the top 4 states in Peninsular Malaysia, namely Selangor, Kelantan, Perak and Pahang, reported around 17% of the total malaria cases in combination. It is worth to note that Malaysia has successfully retained zero indigenous malaria status since 2018 and is on its way to qualify for WHO's certification of malaria elimination for indigenous transmission in 2021. So the highest number of malaria in Malaysia was contributed mainly from P. knowlesi cases notified in Sabah and Sarawak, with a sharp increase for 2017. While in Peninsular Malaysia, the most common type of Plasmodium was P. vivax, followed by P. knowlesi and P. falciparum. However, in 2017, there was a surge in the number of notified P. knowlesi cases in Peninsular Malaysia, despite a decreasing trend for the other Plasmodium species. Malaria is endemic in most tropical countries, and according to the latest World Malaria Report released in December 2019, there were estimated 228 million clinical episodes and 405,000 deaths due to malaria in 2018. The report also declared that Sub-Saharan Africa carries disproportionately high share of the global malaria burden, with 95% of all malaria deaths are concentrated in this part of the world. The most vulnerable groups are children under the age of 5 who have not developed immunity to malaria, and pregnant women whose immunity has been decreased by pregnancy. An unprecedented decline in malaria incidences was recorded during the last decade due to the availability of effective control interventions, such as the deployment of artemisinin-based combination therapy and insecticide-treated bed nets. However, according to the World Health Organization, malaria is staging a comeback in part due to the development of drug resistance. Resistance is primarily seen in Plasmodium Falciparum, which is the most virulent human malaria parasites. Many countries with a high rate of malaria are basically underdeveloped nations, and the disease maintains a vicious cycle of disease with poor sanitation and poverty. And malaria chemotherapy has a strong historical link to natural products. The most successful anti-malarial agents have their origin in plant metabolites. The first successful anti-malarial drugs was quinine, isolated from the bark of the South American cinchona tree. The active principle quinine was first isolated from the bark during the early 19th century. Quinine is an alkaloid, a naturally occurring chemical compound that gives it its bitter taste. These compounds serve as a lead structure in the development of the successfully synthetic anti-malarial chloroquine, which in recent years has fallen out of favor as a result of the development of drug resistance by the parasites. For many years, quinine remained the major anti-malarial drug, but from 1930s and at the end of Second World War, this natural product was largely replaced by a series of synthetic drugs, including 4-aminoquinolines, such as chloroquine and hydroxychloroquine, 8-aminoquinoline, such as primaquine, and folic acid synthesis inhibitors. The quinoline-containing anti-malarial drugs are thought to act by interfering with the digestion of hemoglobin in the blood stage of malaria life cycle. However, again, the use of these classes of drugs have been decreased due to the development of resistance in parasites, especially against P. falciparum. Next is the archemazine. Archemazine was first isolated in 1971 from the leaves of Artemisia annua, a herb that has been commonly been used in Chinese traditional medicine to treat fever. So due to the great positive impact of artemisinin in combating malaria, Professor Yu, who first isolated artemisinin, was awarded the Joint Nobel Prize in 2015. Archemazine has a higher chemotherapeutic index than chloroquine and is effective in chloroquine-resistant strain of human malaria. Archemazine serves as a lead structure for the development of new anti-malarial with improved physicochemical properties. Some of the important derivatives are artesunate, artemether, and dihydroartemisinin. The mechanism of action of Archemazine derivatives appears to involve the heme-mediated decomposition of the endoperoxide bridge to produce carbon-centered free radicals, followed by alkylation of malaria-specific protein, which cause damage to the parasite membrane. Archemazine-based combination therapies are recommended by WHO as the first and second-line treatment for uncomplicated P. falciparum malaria, as well as for chloroquine-resistant P. vivax malaria. ACTs combine an archemazine derivative with a partner drug. The role of the archemazine compound is to reduce the number of parasites during the first 3 days of treatment, which is to reduce parasite biomass, while the role of the partner drug is to eliminate the remaining parasites. However, the emergence and spread of multi-drug resistance malaria parasites in the greater Mekong sub-region represents a threat to malaria prevention, control, and elimination efforts. Therefore, in an effort to combat the emerging drug-resistant parasites, our research team at Ataur Rahman Institute for Natural Product Discovery has established screening anti-malarial assays in search for new lead compounds from natural resources. Supported by computational studies, we hope this research will enlighten some path towards a new hope to fight against drug-resistant malaria parasites. For the in-vitro Anti-Plasmodium study, we utilized the Plasmodium lactate dehydrogenase assays, or PLDH assay, which is based on detection of the Plasmodium intracellular metabolic enzyme, the lactate dehydrogenase. The tested extract or compound will be tested against chloroquine sensitive and chloroquine resistance of P. falciparum. The degree of selectivity of an extract or compound shows for the malaria parasite can be expressed by its selectivity index or SI value. This is important to ensure that the tested extract or compound is selectively toxic to the parasite with least cellular toxicity towards human cells. Further validation of the anti-malarial efficacy of the tested extract or compound will be further determined using suppressive tests in P. Bergey infected mice. Parameters such as parasitemia level, survival over time, hematological, biochemical, and histopathological changes will be observed. And with that, I think is the end of the presentation. I guess we are open for questions.
Hello. Dr. Nufruda is asking about what happens to the artemisinin. Is it no longer works because your work is actually to find some new candidates to treat malaria? So artemisinin is quite potent to treat malaria. So can you comment on that?
All right. As I have mentioned earlier, archemazine, malaria parasite, especially P. falciparum, has gain resistance toward archemazine. They have WHO has reported that they have observed cases of archemazine resistance at the Mekong sub-region. So there are high risks of possibility that this case will escalate later in other parts of the world. That's the reason why we are in search, we are focusing to find a new lead compound which can actually fight the resistance parasites. Does that answer the question?
Yes. Thank you, Dr. Rozaini. Sorry. Thank you, Dr. Rozaini. There's one more question from Dr. Fatima. I would like to ask any potential plant sample that has been screened towards the parasite by our team.
All right, thank you, Dr. Fatima. Yes, in our rings, we basically focus on goniothalamus species, and we also screen not only the extract but also the compound, mainly styrolactones, which have shown potent anti-plasmodial activity towards the Plasmodium strains, especially the resistant strains. And we have managed to validate that inside the animal model, whereby the extract and compound not only give promising anti-malarial activity, but it can also ameliorate the injuries, tissue injuries caused by the malaria parasites.
Thank you, Dr. Rozaini. Anyone else? Any question? So if you don't have any question, the attendance form has been posted, and please fill up the attendance form, and we'll meet you again in another session. Thank you everyone for attending this webinar, and we'll see you again next time.
Right, thank you.